Hello, welcome to the PBM and Tinnitus Lab channel. I'm Dr. Katoyun and this is the third session of the PBM series. In this video, I want to answer the question why PBM seems to work for such a wide variety of health issues. From allergies to pain to slow healing wounds, mood disorders, brain injuries, signs of aging, inflammation, neurodegenerative disorders, and of course tinnitus. As I told you, I believe that it is the future treatment of tinnitus. But how a therapeutic tool can be effective in such a wide range of disorders? The secret is that PBM works at the cellular level, and its main target is mitochondria, the powerhouse of the cells. Mitochondria are at the core of nearly all of the vital body functions. It makes ATP involved in apoptosis, generates heat, and houses its own DNA and transfers calciums and plays an important role in immune system regulation. If you want to remember the functions of mitochondria, you can just remember mighty mitochondria. In mighty, M stands for makes ATP or adenosine triphosphate, which is the main cellular energy currency. I stands for involved in apoptosis. Apoptosis is the programmed cell death. Helps removing damaged or infected cells. G stands for generates heat, especially in brown fat in the infants, and it helps to regulate body temperature via thermogenesis. H stands for houses its own DNA. Based on the endosymbiotic theory, mitochondria have the ability to produce its own DNA, and it isn't dependent to nucleus. And it can produces its own DNA. T stands for transfer calcium. Calcium signaling is very important and regulates the muscle contraction, neurotransmitter release, and cell division. I stands for immunity by producing reactive oxygen species or ROS, and also activating inflammasomes and regulating antiviral responses. Mitochondria have play an important role in immune system. One of the most important functions of mitochondria is producing ATP, the main source of cellular energy. And in this process named cellular respiration, mitochondria consume glucose and oxygen to produce ATP. And there are two byproducts, water, H2O, and CO2. But before explaining the whole process, I should explain I should indicate two important parts of the mitochondrion. You see here is a mitochondrion, and it has two membrane, inner membrane and outer membrane. This is the intermembrane space, and In the mitochondrion, chondrion is a matrix, a viscous fluid. So here is the matrix of mitochondrion. Um, Producing ATP occurs via oxidative phosphorylation, which is a nearly complex process. But I try to simplify it in this image, and so it is. I try to make it easier to remember. You can see these are two molecules from Krebs cycle, NADH and FADH2. These two molecules are from Krebs cycle, which usually happens before oxidative phosphorylation. In the Krebs cycle, nutrients are broken down and their energy save as high energy electrons in these two molecules. So these two molecules are electron donors, and their high energy electron enter the electron transport chain in the mitochondria and pass through these high energy electron pass through the four protein complexes from protein 1 to protein 4 in the mitochondria. It's very important to note that the fourth protein is cytochrome C oxidase enzyme, which is a very important enzyme and is the molecule that detects the photobiomodulation light. So the fourth protein complex is CCO. And the electrons pass through these four protein complexes, losing their energy. And their released energy is spent for pumping positive ions or protons from matrix to the intermembrane space in mitochondria. So this energy released from electrons used for pumping these positive ions or protons to the intermembrane space, and these protons naturally want to come back to the matrix because of the difference in concentration. And in their way back or in their flow back to matrix, they lose their energy. And the enzyme ATP synthase is here to use their energy for converting adenosine diphosphate or ADP plus inorganic phosphate to ATP, which is the main source of energy. You can see that it isn't complex and it is very simple process. Before I explain the effects of PBM, I should talk about two important terms in photobiomodulation. A photoacceptor is a protein or cell that detects the light and starts a signaling cascade. Here a photoacceptor. And chromophore is a part of the molecule that absorbs light within a photoacceptor. So chromophore is a part of photoacceptor protein or cell. One of the effects of PBM is on mitochondria is enhancement of the electron transport chain efficiency. It facilitates the transfer of electrons from the protein complex 1 to 4, and I told you that the fourth protein complex is cytochrome C oxidase and or CCO, which is the photo receptor. And hem and copper centers in CCO are chromophores. So the light of PBM is absorbed by cytochrome C oxidase. One of the effects, one of the important effects of PBM is that in pathologic conditions, NO or nitric oxide compete with oxygen to bind in the oxygen site of the CCO, and PBM release NO and dissociate it from the oxygen site, and the free NO enters to the circulation. Free NO is a potent vasodilator, so it can improve the microcirculation, which is one of the remarkable effects of PBM. And also nitric oxide or NO has anti-inflammatory effects via NO signaling pathways, and so it has it works as anti-inflammatory agent. So if I want to number them, if I want to recall the effects of PBM on mitochondria, at first it activates cytochrome C oxidase. It increases producing ATP. It releases nitric oxide from the oxygen site of CCO, and it helps to create reactive oxygen species, which are very important in healing and also in regulating immune system, and also it can promote producing new mitochondria. As I told you, mitochondrion has the ability to produce its own DNA and to make new mitochondria. So it isn't a magic tool, and now you can understand why it is effective in such a wide variety of disorders and health issues. And now if I want to explain how this helps tinnitus, at first I should explain the mechanisms behind tinnitus, and then I will tell you why I think it can be effective in treatment of tinnitus, and it is the future treatment of tinnitus. And now I want to pay a tribute to Professor Tina Caro and also Professor Michael Hamblin, who are the pioneers in this field of science, and they helped decoding the science behind PBM and its mechanisms of actions, and also the other scientists who have have a very important and helpful research in this field of science. Thanks for your attention. Thanks for watching this video. If this video was helpful for you, please subscribe the channel and comment on my videos. I appreciate your comments, and my next video will be about the mechanisms behind tinnitus. So stay tuned and have a nice time.